Okay, let's wait for 2 more minutes because I see people start joining. Yeah. So by the way, do you want to make the talk more interactive, like people can raise their hands in the middle and ask questions, or we wait until the end? Either way.
Yes, definitely open to interactive.
Sure, sure. I got a comment already. Hi, Sam. Excited for your talk on YouTube. Yeah.
Um, so for today's talk, sort of the title and the theme I ended up settling on within the realm of what I've been working on is trying to drive at this notion of what is progress with this vision I have of knowledge aggregation, where I believe that large language model era tools can be helpful towards automating uh, not only the aggregation of knowledge itself, but also uh, automating the writing of new textbooks, having living, up-to-date textbooks that can take textbook style knowledge that ordinarily is thought of to be on an introductory level and actually be able to get uh, what's normally thought of just the foundational introduction to professional training into the realm of what we can actually uh, be able to convey across professional disciplines. So in my past presentations in the region, uh, I've managed to present this conference talk series on horizontal momentum, uh, basically trying to embrace better communication, collaboration, and training across disciplines. And I can share uh, the actual link for previous presentation that I gave. Let's see here. So collectively, I wanted to reference a few um, past events. Um, so this is in the Zoom chat, and it's on the YouTube channel Bethesda Data Science. The title for that talk was Rethinking Foundations, Challenging the Pillars of Conventional Science. Really trying to look at horizontal momentum as uh, sort of two ways that we can picture um, there we go. So horizontal momentum essentially is this uh, demonstration that academic publishing, for the most part, is this incentivization structure that gets uh, previous work. In order for new work to be accepted for academic publishing, you have to establish that you have built on top of prior work. But what that creates is this paradigm where you need to build on top of prior work, uh, almost settling into agreement without even having room to actually be a disruptor or having a distinction versus uh, what old knowledge is telling us. And what it ends up creating is where you can sort of picture as this vertical momentum, problem space of this ski line trajectory, where the path forward is decided and there's no room to change it. So in one sense, what this creates is room to actually automate and propose a computational framework for the path forward. And what I'd like to propose, see here, this one of the first slides. So in the broader title, what is progress, knowledge aggregation, living textbooks, and the automation of scientific discovery? So what I would like to make a case for is that we can automate away a lot of the delays that are incorporated when we have the bleeding edge of new discoveries from new scientific studies. the cutting edge of replicated science, where we have independent replication across separate research groups, and then all the rest of the delays before that new knowledge ever gets incorporated into professional training resources like textbooks. And I believe we can automate away the vast majority of those delays. And similarly, I'm going to make a case that I believe we can automate potentially a plausible framework of the process of scientific discoveries as a whole. So to take everything that we have data on from decades of biomedical literature, taking a look at the metascience of biomedical research, and ultimately being able to take knowledge as the central data that we're going to model, rather than using conventional data types that might be a direct measurement like quantitative, temperature, or volume. I'm much more interested in modeling knowledge. And the biggest use case I have of this is around this heterogeneous syndrome called ME/CFS. So the name that more people have heard of before is chronic fatigue syndrome. The newer, more scientific name is myalgic encephalomyelitis. So ME/CFS. It's a very heterogeneous syndrome. And what I have on the screen share right now is the poster that I presented at the Medical University of Vienna in November at the Emerge Early Career ME/CFS Researcher Workshop. And basically, this is a condition that is a very, very heterogeneous syndrome. It has very different presentations that varies widely from one person to another. Consistent with most syndromes, it's difficult to sort of distill down the simplicity of what is the core essence of this condition or syndrome, because there's just so much variability and heterogeneity. So the tagline of this poster is how to teach old data new tricks, with the overarching narrative of subtyping development in this heterogeneous syndrome. How to teach old data new tricks is using all the data that's already generated to date, biodata, such as the actual study data sets that will get published alongside publications, as well as the actual publication full text, treating them as text-based data sets. And the whole reason I use the term or the wording, how to teach old data new tricks, is this project, this computational framework has no requirement of generating new studies or new data or experiments. It simply is exercising the translational axis, forming novel combinations of basically existing resources. So instead of having to make a brand new discovery or invention from scratch or reinvent the wheel, stand on the shoulders of giants and deploy existing solutions. Stepping through a couple of the things on the screen share, I'll be returning to each of these in more depth. This poster is definitely rich and has a lot of content in it, and a lot of complexity distilled into it. But alongside it, there's also the GitHub repo that I'll be sharing some links of as well. Can throw this directly into the Zoom chat. And the overarching goal of taking knowledge that we can model and be able to actually see if we can have training data and test data that could be consistent with all the knowledge we had 2 decades ago, 1 decade later. How does all the knowledge we had at time point A plus time point B turn into proving what we knew at time point C? And some of the computational algorithmic approaches of interest for the frameworks that I've been building up are some of what I'll be demoing today, between the GitHub repo that I just shared a link in the Zoom chat right here. Trombone Engineer is my GitHub profile name. And this is a local rag. So I don't have pictures yet on the GitHub repo for the documentation in more depth. But today I can demo on my own machine. It's called a local RAG because on top of being a RAG framework that can pull, RAG is short for Retrieval Augmented Generation. It's an LLM era tool. And it's a framework that's built on top of large language models like ChatGPT or some of the more rigorous and robust scientific tools that could go far beyond what ChatGPT can handle. Um, and it's a local rendition or configuration of it because it's able to run right here on my laptop where I'm filming this and from where my camera view is from today. So there's no requirement of any subscription or cost. It builds on top of free, local source, and local LLM era tools. And the local RAG that I was able to vibe code as of October and put on my GitHub a few months ago here is actually able to summarize biomedical documents, biomedical publications. So overall, the framework that I'm trying to build towards is really saying that we can take basically arguing why we would use free, local, and open source AI rather than the flagship models that are on frontier style models. I would argue that a lot of what we can do with LLM era tools really comes down to not the model choice or contents or training data of the model, but really how you use it. And the next thing I wanted to step through is my background and how I actually got here. So this slide just has some of the more technical aspects that I'll be layering on top of. But for my background, I'm presently principal investigator for DMV Petri Dish as a nonprofit 501c3 local to the DC, Maryland, Virginia region. And I consider myself a systems biologist at heart. So I prefer to specialize my biomedical research on interactions and connections in biology rather than just one domain of expertise like just neuroscience or only immunology. So by really specializing on the interactions and connections, I've really taken an interest in collecting skill sets and wearing many hats and working across many disciplines. So I have degree studies and training across chemical engineering, bioinformatics, systems biology, synthetic biology, and biomedical sciences. So I consider myself a systems biologist at heart, but also both a scientist and an engineer. And what I've been able to abstract away from those dimensions has really inspired a lot of the work that I've been able to take on a lot of the unmet needs around ME/CFS, which is far from rare, has millions of cases in USA alone before 2020 spiked those numbers, with dozens of millions around the rest of the world even before 2020. And I'll reference back to this for when I can talk to in more detail on the nature of not just why, but how this is carried out as a computational framework for subtyping development. ME/CFS is very difficult to distill down to simplistic terms, but we have a lot of data on it. And we have a lot of individual researchers and centers and efforts that have gone into trying to get basically just exercise the translational axis, trying to get closer from basic science research into translational and applied research, bridging science and engineering. So taking a step back, I glazed over some different field names: chemical engineering, bioinformatics, systems biology, and synthetic biology, biomedical sciences. The first one of those I started out in is chemical engineering. And I have one more link I can add to the chat that this is also linked from the same GitHub page or profile, Trombone Engineer. But this is actually a MATLAB coding demo that I wrote in 2015. So it's also got captions and an overlay describing the autistic cognitive experience as best as I could fit into less than 5 minutes, representing as much of the spectrum as feasible. But ultimately, this came out of the studies I did in my undergrad engineering in chemical engineering, where computational chemical engineering that I was starting to specialize into built on top of basically the scale-up science of chemistry. Chemical engineering as a field takes chemistry from the lab bench scale from milliliters or microliters in a test tube up to millions of gallons per hour of continuous production in a factory. And that science of scaling up chemistry can be abstracted beyond just chemical engineering, uh, even on the computational interface that intersects with chemical engineering that I was studying closely in my undergrad studies. Um, I basically learned that there are two dimensions of where you can specialize computational research, computational frameworks that you can deploy. You can take it as a focus of automating or scaling up. And um, so in terms of automating and scaling up, a lot of what happens with horizontal momentum and the problem space of vertical momentum, as I've spoken to in my past conference talk series, um, a lot of what gets focused on creates these peaks and valleys. You can think of it in terms of computational optimization, in terms of uh, basically trying to do optimization where you have local minimums versus global minimums. And there's these highly trafficked problem spaces that get a lot of attention and just become the most familiar. And then there's all the rest, where you might have completely unattempted or not even acknowledged problem spaces. And then you have solution frameworks that either haven't been explored yet or just haven't ever been entertained in a certain uh, problem space. So in terms of exercising the translational axis, it's really a matter of finding novel combinations and not even necessarily a brand new invention or discovery from scratch. And in terms of scaling up versus automating, automation can sort of be envisioned as this way of taking a familiar process and making it more efficient, more effective, something along that axis. So in conventional computer science, you might have a framework that already exists, a problem space, multiple solution frameworks that have been proposed that are highly trafficked, but really not necessarily a whole lot of impact from what gets addressed in that uh, problem space. Even the solution frameworks get fairly familiar awfully quick. Um, so I would argue there's far more impact to make to address unmet needs and ultimately promote the most good for the most people, to make progress and catalyze change uh, in the untrafficked, unattempted problem spaces. So when you're scaling something up or automating a process with computational power, I would argue that there's a lot more impact behind uh, trying something that's never been thought to be possible before. And one of the things I'll be speaking to in more depth of the decision tree of why I have leaned heavily into building with local, free, and open source AI, large language model era tools. Collectively, the tools we have at our disposal today, and not just exclusively LLMs or foundation models even. Um, I lean much more heavily into local, free, and open source because uh, there's a lot more value we can get from exercising the translational axis with existing proposed solution frameworks and even just basic science that has been already published that no one really necessarily knows about because search is still an unsolved problem. So how do we take solution frameworks that have been proposed or even just knowledge? It might not be an engineering applied framework. It might just be basic science that has to be picked up to the next steps to be applied and translated to the clinic, to the bedside, or some other niche to be translated. In order to actually exercise the translational axis, I would argue it's a lot more valuable to either scale up or automate a completely unattempted problem space. And you often don't have to invent a brand new from scratch solution framework. You can take existing knowledge, existing accomplishments, and just make novel combinations. And a lot of what I do in systems biology is I take discoveries and just basic science across the board, across literature, like taking all of PubMed. And I take this ambition of parsing all of biomedical literature from PubMed. So rather than uh, just taking the assumption that it would never be possible or it's never been done, so really have to be proven that you could do something that new and powerful or difficult. I'm starting to build up from the ground with local free and open source AI. And that relates back to the local RAG that I've been able to um, put the code base up on my GitHub, publicly accessible trombone engineer local RAG. And I could demo that today. But ultimately, stepping back through my background, chemical engineering, then bioinformatics and systems biology, systems and synthetic biology, and then biomedical sciences. Um, there are some really powerful parallels there that I've been able to abstract away from uh, what sort of is confined to just one field or often turns into silos into what we can then connect the dots and figure out the next steps of progress and uh, ultimately going back to the question that I have in the title of the whole talk, uh, what is progress? And what is the direction forward? What direction is progress? How do we actually figure out the right way to move forward if we don't even know the the landscape of what people are talking about or what people stand for or what is actually the next step forward? So I believe that LLM error tools can be very powerful towards this vision of knowledge aggregation to be able to map out transparently different two different flavors of knowledge aggregation frameworks where you're mapping out the transparency of what's already been said, whether it's on a factual basis or not. Just being able to map out what people were talking about and the silos that so much information, so much knowledge gets siloed. How do we take those knowledge silos and connect the dots and address the fact that search is still an unsolved problem and there's a lot of progress left to go, but a lot of the solution frameworks that could be very feasible, you don't have to invent from scratch or uh, build from nothing. And even though I was alluding to a minute ago, I'm starting to build up free local open source AI. I'm actually not building from scratch. I am deploying existing solution frameworks that already exist and ultimately mapping out or building up towards finding the transparency of the landscape of uh, trying to answer what is progress? What is the direction forward? It's going to be very subjective and have a lot of individualistic nature to it. And there's different niches that you can specialize that approach into. There can be progress in a political sense or economic. I'm focused today mostly on biomedical research meta science. So there are a lot of silos in biomedical research just as a single umbrella. So a lot of stuff doesn't get connected. A lot of stuff gets siloed and stuck. And a lot of the incentives that exist today don't really facilitate going beyond the silos. We sort of have a propensity to get stuck and comfortable in the bubble of our primary professional discipline. Not even just in science or STEM, but broadly. A lot of professionals interact with structures of what's called reductionism, where a lot of the familiar, uh, highly trafficked niches of the vertical momentum uh, a lot of what we do as professionals just becomes really familiar. Um, so I will also take a step back and say, I have slides that I can share and the resources I started putting into the chat over Zoom uh, between mostly these two YouTube videos, uh, GitHub with Trombone Engineer with the local RAG. Um, And I'm happy to make this interactive as well. So I wanted to sort of lay the groundwork so far of how can we even begin to reach consensus agreement or have any hope of moving forward together to promote the most good for the most people if we can't even map out the transparency of what is progress, what direction is forward, where do we want to proceed? And I think it's time to reevaluate a lot of incentives in a lot of contexts, including directly in biomedical research. So uh, some of the computational algorithmic approaches of interest that I've really spearheaded in my lab with the ambition of parsing all of PubMed. Even though a lot of people would say it's not possible or would never be affordable or feasible, I would add the caveat that it's not affordable if you're stuck on flagship AI models that are going to be uh, basically expensive or charging per amount of data that you have to go through. And with something as large as PubMed, all the biomedical publications that are out there, it's going to be difficult to work through all of it in terms of cost. But we already have the tools to be able to build up from what other people have already built, just deploying open source solutions, exercising the translational axis, taking what's already been proposed and connecting the dots in novel combinations. And a lot of what I do in systems biology is I take novel combinations of software tools and data sets and parameters of how you use the software tool or interpret and process the data. And just by forming novel combinations of those, you don't have to have any brand new data set that's new to the world. You don't have to come up with a new software tool or algorithm. You just form novel combinations, and you can extract additional insights from data, like I do in this framework around ME/CFS subtyping development, without having to actually make any new discovery or invention from scratch. So there's no requirement of generating new studies or data sets or experiments. And the pipe dream in this project, if everything can go according to plan, in this bottom right quadrant, it's synthetic data for now, violin plots. Was working with some other data sets that might be representative of this summary goal. But this specific plot is synthetic data to illustrate the point. What if we could find significantly effective treatments hiding in old clinical trial data sets? So ME/CFS is far from rare, but has 0 FDA-approved treatments to date. And it's very heterogeneous. So in this left half of the violin plot here, the traditional analysis section, all the heterogeneity manifests by saying you have all the individual dots of individuals in the cohort for cases versus controls. So controls are a bit tighter here because there's less heterogeneity than within the heterogeneous syndrome ME/CFS. And it's not a significant p-value when you look at are the cases any different than the controls on the specific hypothesis being evaluated in this p-value calculation. But if you can actually exercise subtyping development and connect the dots between underlying mechanisms and objective biomarkers and ultimately discern responders versus non-responders, there might be a fighting chance to actually find significantly effective treatments where we have effective p-values between these comparison groups, not only between cases and controls, but specifically pockets of insights from subsets of heterogeneous cohorts or populations. So a lot of this that I'm manifesting in the dimensionality of the data analysis in this framework for ME/CFS is actually very parallel to how I see knowledge aggregation broadly, because there's a lot that we can deploy from existing discoveries, existing proposed solutions. There's often a lot more that's out there in published literature, not even just biomedical literature, but broadly, academic publishing wise. A lot of the times when we humans think we've come up with a new idea and that we're the first to stumble upon something, it's usually already out there and published. And just we're either not aware of it or search is still an unsolved problem to such a degree that it's a hard, difficult time to find what you're looking for, even if you do intentionally go out looking for what's been done in your field. And part of the nature of my drive to automate the writing of new textbooks and consolidate everything that's been put out there is there's so much just explosion of exponential increase of new information to the point that many fields, I would argue almost all, are facing this challenge across the board of there's way more information even just in academic publishing alone, let alone all the rest of the dimensions of data and information, that it's hard to keep up. It's hard to keep up with change and what's not true that used to be true. What information is good quality information in terms of how accurate and representative a claim is versus evidence. And ultimately, trying to discern correlation versus causation and this kaleidoscope into directionality. So it's important not only to recognize that correlation does not necessarily prove you've established causation in science, but even if you can start to entertain enough correlations that are connected and significant, can you actually untangle the directionality in complex systems like human physiology? So a lot of what this framework is doing, I'll speed through the flow chart that's here. Phase 1A, 1B, 2, 3, and 4. The tail end, phase 4 here, isn't really hoping to question the validity or accuracy of old analyses. I'm just trying to figure out what does it all mean in context, synergistically together in the big picture. What does it all mean together? What is the ramification of this new finding that a study has proposed? So collectively, this framework at phase 4 is just putting everything into biological context, just figuring out what does it mean in the big picture. Not questioning the validity of the findings, just aggregating everything that's been put out there. And going back to the top of the flowchart here, synergistic, parallel data mining and text mining take all of the existing data that's available from biodata like metabolomics, proteomics, metabolites, mutations, things that biomedical studies tend to really focus on as the data before actually looking at the metadata that's important for data science and database management and computational analyses. There's many manifestations of metadata that become important here that I've learned by helping to host data science competitions and biohackathons. It's very difficult to consolidate what's out there. And even hackathons often struggle to summarize and consolidate across separate efforts. A lot gets siloed, a lot gets just stuck in one incentive paradigm. And part of my mission with knowledge aggregation is taking an algorithmic approach to identifying and maybe automating identification of paradigms, maybe even paradigm shifts, or where we might be overdue to overturn old knowledge and identify paradigm shifts, or just where there's room to question old knowledge. Because academic publishing, like I've spoken to in my past conference talk series on horizontal momentum, really struggles to accept any old knowledge being questioned. And there's that ski line trajectory of the path forward that you can't question. It's stay in your lane professionalism. I'm a this, I can't comment on that, hyper-specialization. And this is the path forward, don't question it. You have to build on top of prior work. But if you don't actually, it doesn't matter whether you establish in that paradigm of academic publishing, whether you have approached reality, or if you're just getting stuck in theoretical. What if the closest approach to reality in that vertical ski line trajectory was back here decades or centuries ago? And then we run into these mentalities of, what do you mean? Why do we have to change our minds? We've known this has been true and rock solid ground truth for decades or hundreds of years. Well, maybe, just maybe, witchcraft is your literal answer. If we prove that this scientific fact was true because of literal witchcraft in the mix of the evidence that was established some centuries ago, maybe, just maybe, it's time to reevaluate and pivot. So the computational frameworks I'm proposing and building up now, especially heavy on the text mining side, that I can get into more depth in the poster in this project soon. A lot of what I'm noticing there's a couple comments showing up in the chat. And I'll also say I'm open to interactive questions. I'll try to answer both the questions that are typed in the chat shortly. there's a lot of room to trying to find 1 second here. Text mining. Okay, so I see two questions here. The first one was a few minutes ago asking about progress versus civilizational advancement. Um, so one of the things I spoke to in the first iteration of my horizontal momentum conference talk series at the ISSS conference in 2023 for the International Society of System Sciences is that a lot of us showed up at that international conference already very accomplished and successful, and mostly across the board aiming to figure out how to scale up success. But how do we actually take success that works at least once and be able to scale it up? Usually we turn to computational frameworks to help us with some of that scale up propensity, some of the benefit of how computers can scale better than human effort. But how do we grapple with the distinction between scaling up success versus progress? And there's this important, huge distinction between what is progress and can we reach consensus agreement on what is the path forward? What is progress? And can we be successful in catalyzing change and progress? And not everyone's going to have the same definition or understanding of what is progress. But part of what's important in knowledge aggregation is being able to consolidate the various effort streams and data dimensions that are out there. So in this project specifically on the screen share, it's very difficult to just imagine that we'll make it there and finish being successful and make enough progress if we remain siloed. So I would argue that we're not going to make it if we stay siloed and just overly reductionistic, overly reductive. Reductionism inherently drives this notion that a question can only allow for one answer. And it's very pervasive. Its impacts really drive a lot of what we do and think about and the limits and bounds of what we interact with on a professional basis in any trade across many flavors of professional work. So one thing I wanted to incorporate, and I have to find the right slide that will be able to demonstrate this the best. 1 second here. Yeah, so let's go to this slide. Okay. So this slide I'm thinking of, apologies, I had a bit of trouble getting my slides to cooperate in preparation for today. Happy to share the resources after today. But there are three algorithmic approaches of interest. Yeah, I think this will do the one. Here we are. Here we are. So I know a lot of the attention and hype is around large language models and LLM era tools these days. I tend to focus and lean a lot more heavily into natural language processing without LLMs or foundation models even. So what this allows us to get access to is the ability to summarize text algorithmically in high throughput without having to risk hallucination, without even requiring any generation of new text. You don't have to synthesize or generate any new text in order to highlight or summarize if you highlight existing text. So finding the needle in the haystack with high throughput algorithms. They're not always GPU oriented the same way deep learning with LLMs tend to be. But often they can benefit from GPU acceleration if you find the right software tool. And specifically, extractive summarization or extractive QA, parts of speech tagging, and temporal knowledge graphs is what I've been wanting to connect the dots to speak to uh, with a couple of the talking points I've already touched on. Um, I believe this last one gives us the best algorithmic framework to be able to plug in a solution or approach to address how does everything we knew a decade ago plus 5 years ago prove what we know today? And if you can't grapple with the temporal dimension of that data, it's going to be a lot harder to grapple with that analysis. So temporal knowledge graphs I've taken an interest in towards actually building a framework that can have the time-gated knowledge, uh, for instance, from PubMed. So how does knowledge from time point A plus time point B prove what we know at time point C? And a lot of what's in these other slides in between, um, one of the frameworks that I like to uh, build within is this three-word phrase of consolidation by coherent corroboration. So both flavors of knowledge aggregation that are um, really heavily leaning on correlation, causation, and directionality of does A cause B or did B cause A? Uh, you might have a stronger connection than just one correlation alone. Often, in order to establish causation, we have to stack many interrelated correlations, overlapped nodes, overlapped connections. Um, and there's a factual, true, and often mechanistic connection in that train of thought. But with the other flavor of knowledge aggregation proposed frameworks, I'm much more interested in just mapping out the transparency, where you might have corroboration that's not necessarily mechanistic or not as coherent as uh, the causation of a scientific process. But a lot of the times, scientists are not immune to the human propensity of believing in something more heavily than we have the evidence to back up, and saying, my work is the greatest. I know I'm right. And a lot of that stems from in science, we learn from meta-science. We set up so many scientific experiments to stack the deck so that we can be correct and validate what we're setting out to look for. So much of what we try to evaluate is a biased validation effort from the get-go. There's a great book I worked through recently called Science Fictions that really addresses meta-science and some of the statistical fallacies that a lot of scientists are guilty of. So addressing this back to the slide, it's important to think about how well do the information streams corroborate each other? Is there any overlap or relatedness, like you can address with cosine similarity in a RAG framework? Is there agreement? Is there consolidation? Is there corroboration that you could consolidate? And how coherent is it? So this is the biggest computational framework I'm building up within is consolidation by coherent corroboration, where I basically think of consolidation as downstream of aggregation, where aggregation is much more ripe for high throughput computational automation and scaled up. You can scale up a lot more progress with aggregation through automation, even if it's a totally unattempted problem space. Consolidation requires curation, manual human effort to curate what we can ultimately consolidate. And there's not a whole lot of either of them that's been done in biomedical research. So much remains silent. So some of it will be mapping out the biomedical research of what do individual researchers or experts deem to be the path forward and direction of progress. Not everyone agrees. Not everyone has the same vision. And within that paradigm, some of the claims, some of the sentences or knowledge really drives home at the causative basis that is true to biomedical research and science as a whole. And some of it isn't. Some of it sides back with the very human nature of assuming intuitively when you see repetition plus consistency that something must be factual. We see it all the time online. I know even myself, if I see something enough times online from enough sources, kind of intuitively will feel like it's true. It's got to be some basis behind it. And scientists are not immune to that propensity within research, let alone beyond it. So a lot of what this uh, framework and approach algorithmically is looking at is mutable knowledge bases. If we want to question old knowledge, if it's the foundation of a skyscraper, it's going to be pretty hard to figure out the right way to pivot. It's going to be pretty hard to adjust the knowledge, the ramifications of that knowledge foundation, of that knowledge base, if any of it is to be overturned. So, for example, there was good science news coverage a couple years ago that one of the foundational pieces of research in the Alzheimer's disease field around the amyloid plaque hypothesis as a central basis or underlying mechanism of Alzheimer's disease. Turns out one of the early studies was completely falsified and had fabricated data. And I've been watching the space and watching the science news coverage. So between the scientists, the science journalists, and just people across the board, no one is really thoroughly taken into account how to consolidate what is across the board in all the different proposed evidence bases, proposed knowledge, proposed conclusions, or even recommendations. A lot of it just remains murky because people are in one silo or not incentivized to consolidate. So it hasn't even really been aggregated yet. A lot of it is still in the unmet needs column for how do we actually aggregate the transparency to map out what people are talking about and what's needed next. So that goes back to what is progress and how do we map out the path forward. And if you sit down and ask the average scientist or enough scientists, you might discover that the siloed nature of hyperspecialization creates this far removed nature of scientists interact with other scientists and other professionals, but rarely the people that are impacted by the work that they're doing in biomedical research. Admittedly, scientists, without being clinicians, aren't structured to give clinical advice, and that creates boundaries that are difficult to navigate for many professionals. But I think it's important to engage with both sides of the fence and not just default to the mentality of I'm at this, I can't comment on that hyper specialization, or stay in your lane, don't look across the fence professionalism. We can still be professional and still promote and establish confidence beyond your primary professional discipline, like I spoke to in my horizontal momentum talks. So let me take a step back here and look closer at the chat here. So that was one of the first comments that I noticed was scaling up success versus progress and progress versus civilizational advancement. I actually do have in my slides directly in my old conference talk series, which I can also spin up here. One of the questions here is about decentralized science, which I happen to have handy right here. So DeSci, for anyone who's never heard of it, is a acronym or shorthand for decentralized science, and it shares a lot of commonalities in parallel with DeFi, decentralized finance. One great explainer video I found is DeSci is basically blockchain technology or crypto for science. So there's different ways of framing or trying to understand what is DeSci. I first approached it as understanding or wanting to see it as a movement of people acknowledging and trying to address a lot of the ways that science and research are broken today. So not just acknowledging the problem space, but proposing solutions, trying to improve the state of science, trying to modernize science. And a lot of the dedicated orgs, many of them called DeSci DAOs, DAO is another acronym, Decentralized Autonomous Organizations. It's the most common org vehicle within DeSci. So many of these orgs are DeSci DAOs, even beyond this box that says DAOs. Um, and there are different niches of what uh, specific DeSci DAOs or orgs initiatives have set out to accomplish. This particular sourced image, I believe this link is still active on Twitter, back when it was just called Twitter. 2023 is when I grabbed this link. So this is somewhat outdated. It's heavily outdated with how fast the space moves. But I wanted to convey that even just as of 2023, when I got this snapshot of the landscape in DeSci, there are different missions, different efforts that specific orgs are aiming to accomplish or set out to try to improve between publishing, like I mentioned, academic publishing, problems in that space and how we could maybe do better, as well as financing, cryptocurrencies, blockchain technologies. And really, DeSci does more than just blockchain technologies as the most well recognized, most familiar cryptocurrencies like uh, Bitcoin, Ethereum. There's a lot more going on in DeSci than just the speculative side of crypto. Um, so I will acknowledge that there are other, it's a more complicated nature to DeSci than just it's blockchain or crypto for science. But that captures a lot of the complexity going on. There's a good explainer video I met someone that made on YouTube that basically gives that punchline. Um, Let me check. I think there were a few comments now that DeSci World with blockchain question is one I see from a couple minutes ago. Um, let's see. I see there's a couple comments that are overlapping here. I'm also open to having this be interactive. If anyone wants to voice a question or reiterate in the chat, feel free to inquire. Let me take a second here to study a couple of the questions in the chat. So that's definitely a good question. Just came in is how to address publisher paywalls on data and dataset silos that are not open due to compliance standards. Um, so compliance is one major keyword. Another is incentives. Uh, and I would argue that a lot of the incentives are due to be reevaluated. Um, a lot of scientists don't know the nature of the heterogeneous syndrome that they research. Not only in MECFS, like I've taken an interest in my lab to research, but just across the board. Uh, a lot of biomedical researchers don't know more than the sort of oversimplified nature of, I guess, one term that gets used is a concept drift. A lot of concepts will use different terms over time or with different connotations from different people or communities. Um, Many people envision a different construct, even from the exact same words or description. And I would say that reductionism tends to fail some of the most complex medical conditions. And similarly, the incentives that we have to question or rethink across the board, not just in biomedical research, but just broadly, a lot of incentives are failing us and just not enough. So sort of the framework I operate within is what happens when we question and make mutable all knowledge? What if we put some confidence score on every little piece of information? What if nothing is rock solid that could just never be overturned because it's too confident? What if we don't know the ground truth or absolute reality? How do we actually iterate around that, simulate or model anything around it? Or how do we grapple with reality without knowing the ground truth? And I've worked enough in predictive modeling, thanks to chemical engineering and even bioinformatics, to know how terrible we humans are at predicting the future. So I put very little confidence into any man-made future prediction, to the point that I've arrived at the philosophy that there are no guarantees. Nothing is 100% confident or predictable. Even 100% chance of rain in 3 minutes is no guarantee. See a question that just came in asking about when I alluded to witchcraft. So in the horizontal momentum conference talk series that is on YouTube, I will put this link back in the Zoom chat in case we had new people. And I can share these resources with the registered attendees after the session ends today as well. What I basically have tried to speak to on my past conference talk series on horizontal momentum is this vertical trajectory, sort of like a ski line, that basically goes this way. And you can't change it. Academic publishing says that's the path forward. Don't question it. We can't overturn or grapple with old knowledge being not the truth, or how do we update our knowledge bases and comprehension if we were wrong, or if we find fraudulent science, or if usually I find it's the former, that we just were a little bit off. And we need to put the findings into broader context of the big picture rather than necessarily question the validity or accuracy of the data or the findings. It's just, there's a lot of complexity to grapple with. And how do we put it all into the big picture in context? When I mentioned witchcraft, it was to this train of thought of this common mentality of what do you mean? Why do we have to change our minds? We've known this has been true and ground truth and absolute fact for decades or centuries. Why do we have to change our minds now? Well, if you look far enough back, enough years, science took a different form. There was a point when alchemy was the state of the best science that we had. I took a great course in my undergrad studies called Ancient Science and Technologies, where we actually dabbled with more than just a surface view of basically the state of science prior to 1400 AD. And there's a lot of flawed approaches that we would like to believe we've just gotten better over time and we've done better and consolidated everything we've made mistakes around before. But a lot of times it gets murkier than that, and it's harder to resolve or consolidate. So witchcraft maybe wasn't considered the state-of-the-art science of the best we could do, but we humans have some bias and some mistakes that get incorporated into even our state-of-the-art science, the best we can do. So if we don't have room to overturn or question old knowledge, that vertical momentum is just so pervasive. All these structures designed and shaped by reductionism. The inherent notion that a question can only allow for one answer, where you solidify a foundation that builds a skyscraper. And it's this vertical trajectory that's a lot like the ski slope or skyscraper, if we want to envision it. The other way to picture horizontal momentum is that it feels like you have to pioneer the space to build up any momentum moving horizontally across all the vertical bins shaped by reductionism. So again, the foundation is rock solid and hard to question. And if we proved that something was factual and that we believed it to be the truth because of literal witchcraft, maybe, just maybe, it's time to reevaluate and not just get stuck in the past, because the past becomes the present. And if we don't consolidate with enough awareness and transparency over the decision tree, uh, yeah. Um, and stigma is an important word. I'm just noticing in the chat here. Uh, Stigma has this parallel with placeholder knowledge. A lot of science, a lot of professionals, I will say even, um, beyond the bubble of your primary professional discipline, a lot of your knowledge is outdated. And this is part of the case that I'm trying to make for why it's so important to automate the writing of new textbooks, or to at least have updated textbooks and overall professional training resources. Not only to have the foundational introductory information that might be changing, or uh, have new, basic, simple knowledge, but also the more complex, harder to distill knowledge that is rapidly changing in every field. A lot of knowledge outside of your primary professional discipline, the secondary disciplines, tertiary, and beyond, um, it's going to be very outdated, and you're going to have a lot of placeholder knowledge. Stigma often acts like placeholder knowledge, where if you don't know better, maybe the only exposure you've gotten to a concept is from uh, what you heard many years ago, or in a joke, or what you thought you learned about something distant to your expertise. The whole reason I try to promote establishing confidence beyond the bubble of your primary professional discipline is because of that placeholder knowledge. It's not always stigma. Sometimes it's just terrible quality knowledge that is literal stigma. Sometimes it's just that our knowledge bases and discoveries and evidence just develop so rapidly that it's difficult to keep up. So if you're a neuroscientist and you want to dabble with immunology, or vice versa, you're going to have to grapple with the fact that your professional training used a textbook that only briefly touched on that secondary discipline, that was probably decades outdated by the time you got the textbook, that you studied and were trained on decades ago. So how do we update those professional knowledge bases, professional training resources across the board to promote better communication, collaboration, and training across disciplines, such as trying to lay out with coining horizontal momentum? And a lot of that placeholder knowledge is really pervasive beyond that primary professional discipline. Where I think the most important thing we can do is have the dialogue, begin the conversation, advocate for the importance of questioning what is progress, what is the direction forward, how do we actually get closer to getting promoting the most good for the most people. And if we're stuck in biomedical research in a way that just remains so siloed, that is just, so stuck on what we do is the greatest and we can never be wrong, well, maybe that's wrong. Maybe it's time to reevaluate. So going to a little bit more depth here before I return to some of the comments. The framework here uses data mining and text mining in the first two parallel stages. Zoom that back in. So the consolidation comes into play in phase two here, using basically the manual curation effort with the data mining, and some automation help with NLP powering the text mining, to structure the biodata as the first column of a human readable table that consolidates all of these statistically significant findings from the entire field into a single human readable table. As long as it's got statistical significance, it makes the cut to get incorporated into this table. The rest of the columns beyond that first column, the biodata, like the metabolites, mutations, uh, the stuff that biomedical studies usually emphasize as the data. Um, Beyond that, a lot of the metadata is crucial. And from helping to host hackathons and data science challenges, I've learned it's not the biodata that empowers or makes or breaks you. It's really the metadata that's going to make or break you. If you don't know the traceability and the transparency of the caveats, qualifiers, and context of the biodata, the findings, uh, the p-value is not the end all. The statistics might not get us all the way there. It might not be sufficient to answer all the questions. So the human readable table takes those biodata types, mostly these five, at least in MECFS subtyping development framework, biomarkers, subtypes, mechanisms, hypotheses, and even recommendations. Because sometimes a finding that has significance or can be significant in the sense of a recommendation might not have a p-value. But if someone's made a recommendation that they've already pushed to all of the public or just general biomedical literature, search is still an unsolved problem. So it might be difficult to find that proposed solution, but it's already out there. And there's so much data in the early layers that still needs to be consolidated into more coherent layers, more streamlined. So this framework is setting out, seeking to grapple with the metadata and the biodata across all of the findings across the entire field, overall to generate steering committee level recommendations for the entire MECFS research field. Grappling with that heterogeneity across many different data dimensions and the metadata and many different software frameworks. And specifically the metadata, I alluded to the caveats, the context and qualifiers. So if the overall narrative that we can distill down needs to be, yes, the study did propose a novel subtype or biomarker for this condition, but you should know that their sample size was limited to 15 people, or they had to design a very specific experimental configuration in order to find this signal, this biomarker. So they're proposing it, and it's got statistical significance in their individual study. On the individual study level, it makes the cut into the table for the biodata column and the p-value that got it there. But the p-value itself and all the other qualifiers of the sample size, other limitations and qualifiers and context, that's all going to be populated into the human readable table as well, so that we can begin to consolidate with the transparency mapped out what is actually going on. What is the dialogue? What's the conversation? What's in the mix? How can we even begin to hope we can reach consensus level agreement and confidence if we can't even know what the building blocks are? So this takes specifically this data repository called mapmecfs.org and the API they provide. When I first made this poster, it was 54 data sets they had available. I was expecting to find maybe a couple dozen candidate biomarkers that had adjusted p-value less than 0.05 as the threshold I filtered for. So the data mining in phase 1A is really just filtering. It's not doing novel analysis. It's not transforming the contents of the data, just filtering down which rows and columns proceed, mostly by that adjusted p-value threshold. So really, when I was picturing there would be a couple dozen potential biomarkers, I was off by a few orders of magnitude. There were 16,339 rows in the data repository that had adjusted p-value less than 0.05. Significant value, statistically significant from the individual study level, where we can begin to place it into broader biological context and figure out the replication ratio. Has this finding ever been replicated across studies? And did they have the same conditions? Did they set out to configure the exact same type of experimental setup? Or did they have completely different approaches? Was one retrospective and another experimental? All those qualifiers, all those caveats and context and limitations, that all needs to go into metadata columns that NLP can help automate to take the findings that are in the papers and the data sets, the data tables, and ultimately surround the context that's in the literature already and just consolidate it into a human readable table. It's going to be a very dense and big human readable table, but the goal is to make it comprehensive enough that you don't have to go back to the original papers to ask a question or look for additional context. Everything you would need to consult to know about the state of that study and the finding is in the human readable table. And then we can begin to look synergistically across studies beyond the individual study level without questioning whether there are mistakes or fabricated lies or anything of that sort, just looking at what patterns are evident. And part of what the early findings in this work uncovered is that the p-value alone, the statistics alone, probably won't get us all the way there. So table 1 here takes just the top 15 most significant adjusted p-values with the molecule name, that every row in the data repository has a molecule name, that's the biodata, whether it's a metabolite, a mutation, it's mostly multiomics, which in systems biology takes this paradigm shift from, for the costing complexity it used to take us to measure just one protein, just one gene. We can now measure all the genes, all the DNA, or all the proteins in a whole sample for about the same cost and complexity it used to take not too many years ago to measure just one. So with omics and multi-omics on top of it, we end up with these studies that find way more statistically significant findings from the individual study alone than we could ever manage to put into biological context and figure out what it means. So we have to consolidate. What does it mean together? What do these findings mean? What are the ramifications? And what could be a plausible understanding or comprehension of, specifically in this table, a lot of these are human genome chromosome number, 11, this one, and then the base pair position. So coordinates and chromosomes in the human genome. I think most of these, if I recall right, are CpG methylation sites, so epigenetics. Still working on streamlining the database management and data science approaches with my lab members to do the due diligence to make sure that if the same molecule shows up in multiple data sets, is it truly an independent replication or is it a database duplicate? If it's just poor database management, it's not nearly as insightful or scientifically informative as if it's an independent replication. And I notice a lot of scientists don't tend to slow down and acknowledge the important distinction between replication in terms of reproducing and replicating findings, especially independently, versus replicating or duplicating effort. So there's this fear that replication is a waste of time, but that's focusing on the second half of that distinction as if it's represented for the whole thing. So it's scary and not novel or insightful or innovative enough to set out to replicate an existing study. But that's assuming that you're going to take the exact same approaches and duplicate effort. If you have a separate data set or data source or separate analysis considerations or methods, it's not duplicating the exact same effort. So not only is it collapsed into the wrong dimensionality to assume that independent replication is a waste of time and not novel or innovative enough. I think it's time to question and try to overturn some incentives and at least begin the conversation, have the dialogue at least going and existing of what is progress? What is the direction forward? Because so many scientists are just looking at the surface level of their silo. and especially struggling to go beyond the surface of all the silos beyond their primary professional discipline or their main niche that they're hyper specialized into. So just real quick, this view of figure 1 and table 2 are different representations of the same histogram data, basically. An actual histogram plot in figure 1 of the replication representation. So the ratio or just total count of how many times the same molecule name is found in this list of 16,000 rows that pass the filter for statistically significant p-value less than 0.05. It's a rough replication ratio of 2 to 1 because there were 8,000 or so rows that were left after I took out all the duplicates of the 16,339 unique or when I removed the duplicates, about half got removed. So about 2 to 1 ratio of replication if we can dot all of our I's and cross all of our T's in my lab and prove that there are no database management errors or mismatches. If it's duplicated data sets and not actually independent replication, there's a huge difference between those and how insightful they are. So we're still working with my lab members towards making sure it's true replication and not just duplication. But the highest number is 16 in both table 2 and figure 1. And it's just in table 2, 5 different thresholds. That's why it's got sort of a diagonal effect visually. So some of these molecules are actually present 16 times in different data sets. And some of the p-values get quite significant looking, or just on paper in an academic framing, they're quite significant. 1.06 to the times 10 to the minus 104. But is statistics sufficient? Or is that yet another silo that's just going to be getting us most of the way there and not all the way there? And I would argue that statistics make for a great start, but aren't going to be the end all, not going to make all the progress or impact for us. We have to go beyond that data dimension of statistical analysis and evaluation. And there might be novel ways to crunch the numbers and come up with different statistical calculations. But we need to make sure we don't get stuck in theoretical land. That vertical trajectory, that vertical momentum, the ski line trajectory of building on top of prior work without any awareness of if you're approaching reality. I would say that's not sufficient. We can do better. And it's important to start the dialogue between professionals of how do we do better. So this framework is sort of my approach towards that. And there's a couple important distinctions in the top right here, just between input subtypes versus output subtypes, basically upstream or downstream of an individual data analysis. How much are you constricting your study's scope as a scientist by trying to stack the deck, whether knowingly, intentionally or not? Many scientists are stacking the deck, trying to make sure they have the best chance of success to measure what we're setting out to look for, and to validate, verify that what we want to measure is going to be possible to succeed and get that statistically significant p-value. But how close to reality, how representative is that? And can we make any faster progress by exercising the translational axis and taking the solution frameworks and knowledge that we can model from other disciplines, other problem spaces, other proposed solution frameworks, and just make novel combinations and model the knowledge with knowledge aggregation. So I also have a couple other demos I can step through, like the local rag itself that runs on my laptop, as well as servers, and the case for why it's important to look at local LLMs. And there's also this framework. So this slide just sort of helps to illustrate the distinction that's important in knowledge aggregation between the what versus the why/how. If we can't reach consensus confident level agreement of what happened, then it's going to be a lot harder to reach consensus agreement and get buy-in towards proceeding on what to do about it, let alone why or how did it happen. So the what here is more focused on what happened, what phenomena occurred, rather than what can we do about it technically. That would be downstream of the what and why, how. Uh, there's a slight distinction between the why versus the how, if you get really technical on the scientific framing and the philosophy. But ultimately, people have different claims or comprehensions of why and how something happens versus what happened. There tends to be more agreement on what happened. And there's also this directionality I alluded to earlier of, correlation is not causation, but directionality is just as important to establish and harder. It comes downstream of, do you have any hint to entertain evidence of causation? If you don't know whether the gut microbiome dysregulation or the brain physiology is causing the other to reflect some strong correlation, it might be more than a correlation. It might have causative ramifications. But if you can't begin to suss out directionality, you're not done yet. So I know that there's a few more questions that have come into a few more comments here in the chat. Slide fitting for what I was alluding to earlier about the mechanistic, causative, logical, or truthful knowledge that you can connect the dots between versus just mapping out the transparency of what corroborates other knowledge, what has agreement or clusters together. And sometimes we have knowledge that's published in like biomedical literature that might be wrong. It might be placing into biological context without comprehending fully what's going on. But it ultimately, there's room to question and reevaluate if we can be transparent enough with the data sharing. Like US federal guidelines now require for any federally funded study, when you publish the paper, you're now required to share the data that you actually collected alongside the publication in repositories like mapmecfs.org. So if we can actually get enough of that data to transparently reevaluate, we can actually do frameworks like this of how to teach old data new tricks and start to look at how much alignment or agreement is there between input versus output subtypes, how much replication is there of separate findings or what's been proposed in the literature. And maybe just maybe if we can be successful and the pipe dream can be accomplished, what if we can find significantly effective treatments hiding in old clinical trial data sets? So I guess just as a takeaway, ramping down for making more room for Q&A in the last 30 minutes. Statistics are often viewed and deemed to be this end-all. Like many fields will often attribute of there's some ground truth or best approach or some things that you just can't question, that there's no room for leeway to reevaluate or pivot. So the p-value alone, the statistical evaluation or calculation, might not get us all the way there. We might have to go beyond the individual study level or the individual theory or hypothesis that we're evaluating. And the targeted data analytics in this framework, phase 3 here, basically takes all of the hypotheses and the biomarkers and subtests and mechanisms and recommendations that can influence and shape hypotheses, and does the same thing of exercising the translational axis, where we don't have to make a brand new invention, discovery, or even form a new hypothesis. just combining novel combinations of entertaining a hypothesis that's never been considered or evaluated in a certain data set or cohort. Just forming all against all combinations, all the hypotheses, all the data sets or cohorts. It might be exhaustively difficult to evaluate all against all. It might even be exhaustively difficult to get all the insights out of one data set when you've got multi-omics where you've got all the proteins and genes and transcripts of mRNA that get expressed, and the epigenetics, microRNAs. Those are a lot of the data dimensions and mapping the CFS specifically. It might be exhaustively difficult to take all these significant findings and place them into biological context. But if we can start to scale up and build a framework of the transparency, begin to have the dialogue and have the conversation around what is the right path forward? Have we done it right already, or do we need to reevaluate? Which of these findings that we view in a mechanistic light might not be iron tight, might not be rock solid. We might have to reevaluate. And even just on the statistical level, we might see findings that carry a very strong value like this 10 to the minus 104 adjusted p-value. But what does it mean? How important is it? How true or accurate is it? And this specifically in table 1, the number 1 hit, this L-cysteinoglycine disulfide. It's a post-translational modification, which technically means within proteomics, it is a small piece that gets modified on a protein. Post-translational just means it's after transcription and translation turns central dogma in biology. DNA goes into mRNA, goes into protein. Translation makes the protein from mRNA into amino acids as the protein chain. Post-translational modification means that you're taking the ability to modify after translation what's in that protein. So you've got the 20 main amino acids that make up most building blocks of proteins, most of the content in them. Cysteine is one of those that's in the name, and glycine as well. So this is actually connecting between two adjacent amino acids and making a disulfide bridge or bond between them. And this was possible to isolate in the current state of the methodologies in a study, had a very strong p-value in cases versus controls. But what does it mean? Does it have more value than just the theoretical, computational, academic framing? Or do we need to go beyond the individual study level, beyond the primary professional discipline, and squash more silos? That's from school. Expected for screen. Checking the chat again. I see there's a lot of activity now in the chat. Also, feel free to unmute if anyone has a specific question at this point.
I'll make some comment. I made a query early on. I put a question mark there: progress versus civilizational advancement. And I really try to say that progress, scientific and technological progress versus societal progress aren't the same thing. And it's very important to distinguish that, because the societal progress is critical, because that also could define the continuity of human species or its own extinction. That's the point I was trying to make. And the other point which I made also about the clean knowledge and the noise, I was referring to AI and digital systems. I mean that AI could contribute to advancements in human knowledge by developing what I call clean digital knowledge. But on the other hand, AI also is contributing for confusing noise. noise and digital systems are generating tremendous amount of fake knowledge. I wouldn't call knowledge, fake noise, basically. And that has got a negative impact on human progress, particularly in science and technology too. And finally, my comments about accessibility of knowledge. The current levels of advancement in science and engineering and in knowledge in general is, I consider, as multi-generational human knowledge, for thousands and thousands of years. We can't say that AI and all these technologies that we see today, it just come over the last 200 years. They are thousands of years. That we have to recognize that. And accessibility is a problem which I see that the digitalization could prevent even the generators of knowledge to access their own knowledge. I'm aware that researchers that cannot access their own papers. They have to buy own papers. I've written technical papers that I have to buy now to access them. So accessibility is one problem. And another point which I would wish to hear from you too is the need for the physical books. If we rely fully on the digital or digitalized storage knowledge, there's a risk possibly that if we don't have physical books, that we may have a catastrophic impact on human evolution. So that's all I want to say. Thank you.
Sure. I mean, certainly, some of the frameworks I've been trying to build up with local, free, and open source AI, and especially trying to lean more heavily into natural language processing without LLMs or even foundation models, so you can circumvent the hallucination risk by not even having that propensity at all. It's important to be able to read and parse large volumes of text with automation help, as well as to generate or handle grappling with what has been generated with Gen AI. In terms of trustworthiness, truthfulness, how representative and accurate a claim is, trying to fact check AI, fact checking LLMs, it's a difficult task. And there's a lot involved in it. There's a lot more than just, is there a source? And the bar is pretty low with ChatGPT, for instance, has fabricated 100% of the citations I've ever asked it for. It's gotten a little better over the years, but I still worry that it's the biggest yes man I've ever heard of. And it's just figuring out how to tell us what we need to hear or believe and not actually telling us what's truthful. It just is so compelling, convincing that it just seems like it's a trustworthy source. But there's so much more involved in seeing how factual and true the sources are and how representative. And you can take a statistic that is accurate and misrepresent it fairly easily. Even scientists can be guilty of it. One thing I'm working towards launching, I don't have in the slides here today, but hopefully we'll be launching fairly soon, is what I'm calling the Magic Theory Pitch Contest. It acknowledges that a magic theory is sort of the extreme end of a personal theory or pet project. On that extreme end, it's when it's treated like it's just like magic. You don't have to question it. Just everything in the universe revolves around this one thing. And every human has a different propensity of what that singular theory or thing might consist of. But the Magic Theory Pitch Contest acknowledges that even scientists are not immune to that human propensity to frame the world and our understandings of textbook style knowledge that might not be rock solid, might have to reevaluate or pivot or adjust some of that knowledge. And the Magic Theory Pitch Contest says, I hear you, that you think or believe that your theory or comprehension is superior. Prove it by pitting the theories against each other and saying, justify that whether you use AI or your expertise or what have you, be able to justify that your train of thought and rationale makes your understanding superior and begin to compare it to the other theories in the big picture and how they're all interwoven together. So it's important to look synergistically and not just stay confined into the silos that are so comfortable of, well, I answered this question. I came up with the one answer that's available, that there's room for this question, because that's just too reductionist. There's way more room to make progress and do better if we can squash more silos.
Thank you. You have answered my questions. I appreciate it.
I see Terry has added a couple more to the chat. Did you have a question you wanted to focus on?
Well, I like your presentation. Your goal, just some small quotes, like saying give quotes summarized by giving quotes of the actual facts from the actual quote. That is so important because, like you say, ChatGPT is just a hallucination engine, and people do not understand how dangerous it is. It is a very, very sloppy compressor of data. So it creates dimensionalities that have no correlation with reality whatsoever, but they sound good. You know, it talks a
It's so
great
good at being a
talk.
yes man. It sounds
Yeah,
like
it's
it's telling the truth, even when it's
ingratiation.
never telling the truth.
Yes, ingratiation. And also I've seen really smart people fall for the ingratiation line. In fact, one person who's an expert who's doing consulting business on AI, I saw a bot go after her and give this incredibly ingratiating story about all she said, and all it was really saying was saying that yeah, use cross product, explore the combinatorial space and you'll get out of the box. That's all it was really saying. You could have said it in that one sentence, but it made it sound like the gross grandiose idea ever invented in the entire history of humanity. And you could tell she was starting to fall for it because it was so complimentary. And I remember I put a point there, and she listened. She listened and said, be careful. These things they'll suck you in. They'll suck you in like a drug. And you see these stories about the genuine AI psychosis. The businessman who goes out into the desert every night so he can wait for the alien spaceships. This guy was a perfectly rational person for 40, 50 years. And he got into this interactive loop in which he's just bouncing his own ideas back at himself. And that's how you don't want to do it. So that single point of advice, get the quote. Get the quote. That's your summarization. Don't do this thing where you let it sloppily schmooze it all together, because that gets so dangerous so quickly. And it's hiding the advantages. There are some good, powerful things about the access. Like you say, it crosses over, it finds things that a human cannot possibly find. And I think, and one of my comments, I think you saw it there. I have a suspicion that the correlation of the DNA tray that underlies biochemistry is contributing to the rationality of LLM applications in that domain, because there's a reason why those molecules show up across very different domains of life. And it's because they work. They have a value and effectiveness, and that puts a constraint on it that I think is kind of missing some other things. So I think that's it's an area where it just feels safer to use some of the LLM. You try to apply that society like ChatGPT does, and what people are saying on Reddit. And of course, it's a disaster because there's not enough consistency in that data set. There's still some reality there. There's still some some good ideas to ferret out, but the noise ratio gets so much higher in cases like that. And it's interesting. But I've always liked the idea of computers are good at memory. They're good at search. They're good at storage. They're good at very, very fast processing. They really stink at understanding, and we don't quite understand why they stink so badly at understanding, but they certainly do. And so your framework seems to emphasize just that point: get the human in at the right spot. So that's I like that. That's a nice feature. And I have talked too long here.
No, that's quite all right. I was hoping to make it allow more room for interactive. I appreciate that people put comments into the Zoom chat. because I think it's ultimately about a dialogue. If we just stay complacent with the comfort of our primary professional discipline that operates like a bubble, the dialogue gets just cut off too often. So in one sense, progress that I was hoping that I set out to try to accomplish several years ago with just the start of my horizontal momentum conference talk series is I found something at the DSI London conference in person that I managed to attend 2023 and 24 in London, largest gathering of DSI around the globe, at least as of those 2 years. First 2 years they held it in DSI London. I noticed there was this problem space that no one was even talking about. The conversation wasn't even started, wasn't happening. So how do we make progress if we can't even talk about it or acknowledge or uncover it yet? And so much of what we can do is undiscovered public knowledge. That's really inspired a lot of my text mining passion in biomedical literature specifically. Don Swanson had really pioneered this. Basically found disparate literatures that had overlapped knowledge bases. I think it's the last slide I have down here, actually. Yep, right there. So it's a wordy slide. I can share all these after today as well. But he basically took manually, before having computational frameworks to help automate it for him, he basically found that fish oil and Raynaud's syndrome both describe very overlapped human physiology. And it's unique versus contexts that don't involve either of those two niches. But the two disparate literatures didn't know about each other, hadn't referenced or commented on each other. So he basically said, we have undiscovered public knowledge. It's already public, it's published. But no one's ever connected the dots. No one's ever entertained the possibility to question it and look synergistically. This was the first time I presented this, made this slide in 2023, first of my horizontal momentum conference talk series. And I used Perplexity AI to look up information about text mining use case examples like by Don Swanson's pioneering in the field, to then do more text mining by Perplexity AI about text mining. So I found it to be a beautiful meta example of what you can do with computational frameworks and scaling up and automating solution frameworks that haven't been attempted or have been neglected and have a lot more room to make impact.
The cross-disciplinary patterns, these are something, like I say, the siloing completely masks these. And yet you see these just powerful patterns repeating, which in a sense is a subset of the whole concept of mathematics. That's what mathematics is about, that you find the patterns that underlie these disparate areas. And in doing so, you find out, oh, even though they seemed unrelated, there is a connection. And usually that connection, the farther apart the fields are, the more profound that connection may be if it's real. And of course, you have to, it could be accidental in some cases, but often it's not accidental. Often there is a reason why these patterns are repeating. And as you know, these are things computers can do far better than humans. We don't have the capacity to do that. And this is, Sam, I don't know if you know my background, but most of my life, my professional life was spent on developing, getting funding for AI and robotics. I always tell people I knew I knew LeCun before anybody knew who he was, so they had the funding was an issue back then. He could have disappeared at one point. And the idea that Jan LeCun could have just disappeared is shocking now. But I had people come in the hallway and say, you shouldn't be funding this guy. It's old fuddy-duddy ideas. It doesn't work. And you look at the history since then, you go like, wow, how could this happen? So
it's one of the common, like, powerful incentives of all the momentum I'm trying to allude to. The vertical momentum problem space and how much it feels like you have to pioneer the space to build up momentum moving horizontally against all the resistance of reductionism in the vertical shaped bins, as well as just people that give resistance of, what do you mean? Why do we have to change our minds? Why do we have to be open to that new knowledge? Because
How do you
it's
feel?
too
How do you
foreign.
feel
It
about
doesn't make sense until you have enough momentum. Most people, it won't make sense to them yet.
how do you feel about the politics of it? Because one of my comments I mentioned the case of what's called string theory now. It was called superstring theory. I've talked to people in that community. They thought I was making up the history. I had to quote the actual statements from people who took the proton string theory and made the superstring hypothesis. And that community is so devoted to that that they actually rewrote their own history. And to the point where people would accuse me of making stuff up. And I said, no, here's the quote, the actual quotes about how this happened. But one of the things I noticed on that, even though you can make an extremely succinct argument and said, yeah, you made it all math when actually it was particles and forces, which is exactly what happened. And then you made the particles and forces, you dropped those and scaled the math 20 magnitudes, orders of magnitude. So you get this thing where people have become very devoted to all that as an assumption. And you have a lot of money involved. It's partially just the ideas, but there's also a lot of money because people are funded, entire departments are funded around it. So I don't know if you've addressed the political side, but there's an interesting political side to these things
But
that can
also,
create
a lot of the times when people will try to blame or acknowledge that money or capitalism must be the bane of existence or the reason for a problem, uh, again, the what happened versus why or how it happened. It's harder to prove. It's harder to find consensus confidence level agreement on is that really why this happened? Will we ever know why or how this happened or how we got here today and through all this complexity, all these interactions and trajectories? I tend to find that ego, overconfidence, especially in science, is just as much of a perpetrator as money for the downsides and the drawbacks of, being a double-edged sword and explaining why we haven't made more progress yet or why science moves so slow and why humans infuse so much bias and mistakes into our work. That bias, that ego, the overconfidence of I need to be right. I have built up so much momentum, so much effort I poured into this. I can't be wrong. I can't afford to be wrong. And that can expand beyond I into we, the institution or organization or lab or what have you. And it's a powerful force. I would argue as powerful as capitalism in terms of downsides and inhibiting progress.
I think they cooperate with each other because you wind up with people, and I know of cases of this, of a person literally sending a letter to a physicist who had dismissed string theory, superstring theory. And basically it was a threatening letter. It's saying you're upsetting think of our children. It actually said something like that. It said like think of the children, think of who you're hurting here. And the letter didn't give a squat about whether it was true or not. It simply said, in this case, they knew something was wrong. And that's a distinction. When people know that it's wrong, and it's not just arrogance, they know it's wrong, but they say, but think of our kids, think of our families, think of our careers. And so you get these complementary threads weaving in and out of each other. But the arrogance side, especially like in the early 2000s and around, I think it's a little bit less now, but there is a thing that I would call continuum mathematics devotion, that no matter what physics says, no matter what any experiment says, the universe is a continuum mathematical structure. And I believe this. So I was much a part of that for most of my life. I'm going like, yeah, but at some point, this goes the thing: is it math first or is it physics first? And I've gone into the camp that said, well, if you don't have an actual physics and objects and a test thing, you can't measure it. So yeah, you can construct the math, but you need to get back to the physics at all points. So, but it at least produces incredible confidence that, all of it is, if you just get that right combination, everything will magically fall into place. And I
Well,
like
there's a
the idea
huge
of
gap
experiment.
between in theory versus in practice or reality.
Yeah.
And academic publishing, getting stuck in theoretical land or never validating or verifying in the real world what you're claiming computationally, theoretically, academically, that's not making the most progress. That's getting stuck in silos, but it's pervasive.
But it feels good.
Yeah.
People like silos. They love how comfortable it feels. And of course that's
Silos
the
and
problem.
bubbles.
Silos, yes. Yeah.
Silos and the
I'm gonna be
bubbles, like your primary professional discipline that functions like a bubble.
I'm gonna be quiet now because other people I'm sure want to talk. So I'm gonna very good talking.
So I have a question, Sam. You talked about representing knowledge as graphs. Do you have some preferred tools that you use to model knowledge as graphs?
So of the main computational algorithmic approaches of interest, um, there's a couple preferred approaches more so than specific tools. I'm still testing and benchmarking a handful of the ones that I've had available. But essentially, um, temporal knowledge graphs specifically, the best resource I've found, if I can remember the YouTube channel, the name of it, I might have to pull it up and double check later. Um, it's one of the first ones that comes up for knowledge graphs. I find it to be the most insightful for temporal and overall for knowledge graphs. Um,
Yeah, are you talking about like a commercial product or
uh, I think it's a company name is their YouTube channel name, just spacing
something
the
like
numbers.
Neo4j or something?
I believe that's the one I was thinking of. Yes.
Okay.
I found they have the most insightful overview videos and detailed technical videos. Um, haven't really found like a textbook that has it, but once again, that's making the case even stronger that we need to have new textbooks and consolidate all this rapidly changing knowledge and evidence into new textbooks.
That's awesome. Great. Second question. Have you looked at some of some of these alternate disciplines for causal discovery? Um, specifically, uh, this gentleman, I think his name is Judea Pearl, published the Book of Why, which is a fascinating read, nicely loaded with lots of statistical paradoxes. But he talks about, everybody knows that causality or a correlation isn't causality. So where does that leave us? We need some other tools to figure out whether we've discovered a causality. Are you doing anything in that area?
The closest and most overlapped of what I've worked on is working through some similar sounding books. I could try to make a list of the ones I can recommend the most. Uh, there's a handful of resources I'm aiming to update on my GitHub profile fairly soon, including the local AI stack that I recently got fully built that uses a lot of the steps I've been speaking to today. Fully local, free, open source. Um, so keep an eye out for Trombone Engineer on GitHub, hopefully within the coming days to have that updated with documentation and instructions on deploying what I've managed to build and deploy on a pipeline level. For the books that overlap the most, uh, Science Fictions, I believe I mentioned at least once, as title. I think it's a fairly recent book. I worked through the audiobook just a month or so ago. And uh, Nexus by Yuval Noah Harari is all about information networks and just information broadly. Uh, and it's not inherently about science or meta science, but I certainly found it to be hugely useful towards knowledge graph modeling and understanding the ramifications of how humans grapple with information over history. And Yuval Noah Harari is a historian by training, so he puts a great historical lens into that book. And there's also the Serendipity Mindset was another book that I really appreciated. I worked through recently. Probably two or three others I'll try to add to the shorthand list on my GitHub shortly. Serendipity Mindset just addresses, I think the tagline was the science of creating or finding good luck. And the main train of thought was just how so often when we attribute scientific discoveries that weren't the most intentional as just being pure accident or lucky mistakes or just dumb luck. Ultimately, the takeaway message I got from the book was the importance of resilience and resourcefulness. If you're not ready to lean into the next opportunity that might come your way, you'll never be able to take advantage of it, even scientifically or professionally. It might be something that happened that you didn't plan for in the biological lab. It might be a mistake or just unexpected occurrence in an algorithm or simulation. You just have to remain open-minded that in theory and in practice, it still has a huge gap between the two. And if we're always looking for what reality must be in theory, we're going to be blind to what's actually in reality, in practice. So hopefully that addresses your question somewhat. But definitely the books I've worked through strike me as the most related to that.
Yeah, it's good to know there are other titles out there. Thank you so much.
Yeah, I'll definitely have to check out the Book of Why. Wrote that down.
Yeah, I just like to. I read that book cover to cover. It's a wonderful read, and especially the statistical paradoxes. Um, it's a great place to start. So
There's also, what is it called? Fisher's Paradox. Uh,
yeah, it's probably in there. I mean, he actually goes through probably a dozen of the classical statistical paradoxes. I think one's actually called Simon's. I could be wrong about that, but it's
there's a
fascinating how
book.
statistics can dramatically get things wrong.
Oh, yeah. Again, it's not the end all. As often as we'd like to believe, it can be that simple. A great engineer once taught me that it takes a lot of innovation and creativity and just effort to engineer simplicity. So we often assume that complexity and simplicity are in a linear, singular direction. But you can build up complexity from simplicity. You can engineer simplicity from complexity. And just saying that we found the answer and that we're comfortable with what we believe is accurate and correct might not be the end all. We might have to readjust, reevaluate, and pivot. There's a lot of progress left to go, and I think we could definitely do better as far as professional productivity and efficiency if we just keep an open dialogue and promote uh, more confidence outside of your primary professional discipline, whether that be statistics or biology or quantum computing or what have you. Oh, that's the title of the book I was thinking of, Bernoulli's Fallacy. So it's about Fisher, uh, Robert Fisher, and statistics. It's a much more technical read. I have not finished that audiobook yet, but Bernoulli's Fallacy is the first two titles of the first two words of the title. It's Bernoulli's Fallacy, Statistical Illogic, and the Crisis of Modern Science. Definitely an interesting read, much more involved than the average book, but seems well written. And there's a couple books in that vein of uh, reductionism uh, in art and brain sciences, Bridging the Two Cultures is a book I finished. The Structure of Scientific Revolutions, Thomas Kuhn. Uh, there's another one, The Knowledge Machine. I haven't started that one yet. How Irrationality Created Modern Science. I'm very interested in trying to figure out how to modernize science because I think it's hard to keep up. It's hard to stay up to date with the changing world around you. And I find that a lot of scientists are more open to rapid change and being up to date and on the cutting edge within their work than within their professional interactions or the scope of how we modernize the rest of science, the rest of professional life, and the rest of human life. So I see one question that I believe was in the chat a few times about has there been any effort to data mine foundational research findings in certain domains that for whatever reason never made it into applied research, and is sentiment analysis also being used? I do have some interest in the latter, in sentiment analysis. I just tend to treat it as not the end all of the only way that you can have power with text mining. But I don't know that there's a single unified effort or any specific examples of work that looks at the foundational research findings and try to data mine them. But I, for one, think that we should put more effort and more momentum into it and have more open-mindedness to question old knowledge and maybe overturn old paradigms, maybe even automate the identification of paradigm shifts, or transparently listing out and mapping out all the paradigms, and maybe the strength and confidence score of paradigms, and see what actually can be corroborated the most confidently and what's less coherent.
Yeah, that's all the question we have. Oh, Terry, you have one more question. Terry, are you aware of any specific examples where the complex portion chemistry or methods of pre-Christian religion may have found a verifiable effect that we now recognize?
Yeah, so the course I took, my undergrad studies in ancient science and technologies, actually shed some light on this. There was even a documentary piece that was recently put out when I was doing my reading in that course. I wrote a little report on ancient neurosurgery within my course. And some of the reading I bumped into, some of the documentaries, made it pretty clear that we haven't necessarily gotten better or more productive or successful over the centuries. There have been discoveries and knowledge that we've lost or lost touch with. Sometimes it's even documented where we can fish it back out and uncover that we lost touch with something that does better than what we're using today. And the most concrete example I remember wasn't an herbal medicine or a drug or supplement, as we would frame today. It was actually surgical equipment, obsidian, surgical, spacing the word, scalpel. So if you use a scalpel that it has material made of obsidian rather than stainless steel, you actually have better outcomes of scarring, surgical wound healing, less scarring from obsidian. And we lost touch with it. And there was one surgeon who found this old historical text, documents that they found this centuries ago. I think it was over 2,000 years ago in this case, ancient Egyptian medicine, if I remember correctly. And it can do better than what we think we've come up with today. So sometimes we have to be open-minded to that human tendency of overconfidence is not always leading us in the right direction. Sometimes we're very off, very wrong. And we just need to have a more open dialogue and squash more silos.
I believe that's actually become a commercial technology now. They've resurrected the idea of glass scalpels. So it has actually gotten some use, I believe. I know I saw an article about that, about using such devices. So much information has been lost. So much.
Yeah, and a big interest I've taken is in the biomedical research meta-science of figuring out what is progress, what makes the most effective, time efficient, or money efficient effort, and what are the best outcomes. When do we lose touch with what used to be the cutting edge that we just don't even entertain anymore? And trying to automate that search and discovery and communication to transparently map out the best path forward, the most productive most successful or most fruitful way to approach science.
Yeah, has anybody ever picked up a shattered piece of obsidian like out in Montana? And it's the definition of stupid sharp.
I mean, from what I've read, I can believe it.
And I must say, it doesn't do much good for bicycle tires.
Can also believe that. Unless you make the tire out of obsidian.
I had a distant relative who made arrowheads out of chert, out of flint, which is very similar to the obsidian. And I was watching him make them once, and then he started pounding on the edge of it, and I asked him what he was doing. He said, I have to dull it because every time I do this, people take their thumb and go and cut their thumb to the bone because they just couldn't believe that something like that could be that sharp. But they don't realize, it's glass. People do worry about glass, but they can underestimate the ancient technologies. Great talk. Great talk.
Thank you.
Yeah, I got a good comment from Maya. Really great and interesting talk. Thank you so much. Please share any resources you can.
Yes, thank you, and happy to share. Ideally, I'm hoping I'll make it onto my GitHub as links and files fairly soon, and happy to share with the registered attendees after today as well.
Sure. Thank you. I will forward to them.
Excellent. And yeah, just thank you everyone for your time today.
Thank you. See you next time. If next time you have more talk, just let me know.
Excellent. Thank you.
And see you next week, Ron.
I'll be there. Thanks.
Yes. Thank you for coming. And you're welcome to come back, Sam.
Thank you.
See you. Have a nice day.
Take care.